Welcome to this episode of Mentors at your Benchside, the podcast giving you advice, tips, and tools for getting the most out of your research. I'm Adam Pawson, and today I'll be talking to you about two of the most common tests used to compare two data sets, their differences, and some of their assumptions. What is the best way to compare two sets of data, and why would you ever need to do it? Selecting an appropriate sample size, calculating the efficacy of your results, and publishing your work may, depending precisely on what you do, rely on statistical comparisons of data. Comparisons have to be fair, accurately represent the data, and show if what you think they show is statistically significant. When comparing two sets of data, you have to make decisions that dictate how you will make the comparison. The first decision is based on how many data sets you want to compare. When you are comparing two sets of data, you have two main options. These are the student's t-test and the Mann-Whitney U-test. Let's learn about these tests and when they apply. Firstly, the student's t-test. The student's t-test, or t-test for short, is the most commonly used test to determine if two sets of data are significantly different from each other. Interestingly, it was not named because it's a test used by students. In fact, the student's t-test was created by a chemist, William Seeley Gossett, who worked for Guinness, the beer company. Gossett used the pen name student to prevent other breweries from discovering Guinness's use of statistics for brewing beer. Who would have thought that statistics and alcohol go so well together? To perform a t-test, your data needs to be continuous and follow the normal distribution. That is, the data are distributed evenly about the mean. Plus, the variance of the two data sets needs to be the same. The t-test comes in both paired and unpaired varieties. In general, most data in biology tends to be unpaired. If you're not 100% sure whether your data is paired, err on the side of caution and assume it isn't. You can use an unpaired t-test on paired data without negative consequences. If you use a paired t-test on unpaired data, you can get a significant result when there is actually no significance and obtain a so-called type 1 error. Number 2, the Mann-Whitney U-test. The Mann-Whitney U-test, also called the Mann-Whitney-Wilcoxon, or MWW, or Wilcoxon rank sum test, or the Wilcoxon-Mann-Whitney test, is used for unpaired samples and is a non-parametric test. That is, it makes no assumptions regarding the distribution or similarity of variances. Therefore, it is less powerful than the unpaired t-test, but you can be more certain that the differences you find between the data are real. The Mann-Whitney U-test is performed by converting your data into ranks and analyzing the difference between the rank totals, providing a statistic, U. The lower the U, the less likely the differences have occurred by chance. Determining if something is significant with the Mann-Whitney U-test involves using different tables that provide a critical value of U for a particular significance level. The critical value varies depending on the significance level chosen, as well as the number of participants in each group, which is not required to be equal for this test. So that's it for the two most common statistical tests used to compare data sets. Check out the episode description for links to related articles and resources, and don't forget to subscribe to the podcast to get more help and advice from mentors at your bedside.